Okay, so good morning, everyone. Thank you for coming. My name is Delroy Ph.D, and today I'll be talking about a context-driven subgraph model for literature-based discovery. I would like to acknowledge and thank those people joining us remotely by GoToMeeting, and we do have several folks joining us as well live on Ustream. We thank you for your interest and for your attendance. So today, like I said, I'll talk about the context-driven...
Could you be a little closer to the mic?
Sure.
So distant.
Sure. I'll stay closer. Right, so today we'll talk about a context-driven subgraph model for literature-based discovery. And just to put into perspective what this talk will be all about, in 1866, Austrian scientist Gregor Johann Mendel had explored the research question of the inheritance of traits in peas. He conducted over 10,000 experiments from which he observed that the inheritance of traits seemed to extend beyond the immediate parents at several generations in the lineage. He was able to explain that the inheritance of traits was as a result of random selection of what he called dominant and recessive factors. When two recessive factors randomly recombine, then we observe features in the generation, in the lineage, that were not present for some time. He went on to put forth what is now known as the Mendelian laws of inheritance as a theory that essentially states that inheritance of traits in plants, it's governed by this random selection of these dominant and recessive factors. In 1903, American scientist Walter Sutton conducted experiments in which he explored the research question of the mechanism of cell division in living organisms. And he conducted experiments with the embryos of grasshoppers, and he found from his observations that in fact it was chromosomes that occur in pairs that split and then randomly recombine. German scientist Theodor Boveri made similar observations working with the embryos of sea urchins and roundworms. Together they put forth what is now known as the Boveri-Sutton chromosome theory, which in fact states that it is chromosomes that are the basis of genetic inheritance. Sutton and Boveri were able to link the observations in plants to that in living organisms, thereby explaining the mechanism of interaction for the inheritance of traits. These two scenarios, and among several others, they capture some general aspects of what we know to be the science of making discoveries. Typically, there is a human who has a research question. The human will conduct experiments. These experiments will yield observations. From the observations, the human will then derive explanations. If the explanation is consistent across a sufficient number of cases, then the human will put forth a theory. In today's technological world, with much of our data in digital format, with information processing systems being developed to facilitate processes such as literature-based discovery, it's important to understand the role of these information processing systems in this general framework of the science of making discoveries. I share the view put forth by Don Swanson that the purpose of an information processing system is not to take a series of observations, process these observations, attempt to arrive at an explanation or a theory, but rather the purpose of the information processing system is to process a series of observations, find promising links, and present those to humans, who will ultimately then be able to reason on the data to try to arrive at an explanation or a theory, as the case may be. I believe this is true because humans are better able to process and appreciate context more than machines. For example, in the case of Mendel and Sutton, it took the human acumen, the insights of Sutton, to really be able to link his observations in the cells of living organisms to plants, experiments conducted some 40 years before. And so ultimately it is humans who will make the discoveries, not so much the computational system. The obvious question then is, how do we develop a computational system that will take these observations and provide these promising links? What are these promising links? The purpose of this PhD dissertation is to present a system, an information processing system, that takes a series of observations, processes these observations, arrives at promising links, and presents those to humans to facilitate the process of making discoveries. This is consistent with my thesis statement, which says that an information processing system that leverages rich representations, which we will show, of textual content from scientific literature based on implicit and explicit context that we will discuss can in fact be effective, an effective means of facilitating literature-based discovery. The motivation for this research, it is not inconsequential. Many of you will remember that in 1999, the pharmaceutical company Merck had launched the anti-inflammatory drug called Vioxx, the brand name for Refecasive, to treat arthritis. Vioxx was highly touted. It was said to be much more effective, a stronger pain medication than naproxen. At the time, that was the brand name for that was Aleve, and Vioxx was also deemed to have less severe side effects. Unfortunately, what Merck did not realize, and this information started becoming public in 2002, is that Vioxx caused an increased risk of heart attacks. In 2004, they did a clinical trial that confirmed this to be true, although they attempted to sidestep the issue by claiming that their faulty results could be blamed on the control. A subsequent publication proved or showed that this was not true, which we'll talk about. Several bad things happened as a result of this. One, Vioxx was voluntarily removed from the market by Merck themselves. They were subsequently sued in 2005 and settled a lawsuit for $254 million. They also, by 2007, were sued by up to 50,000 people, and they agreed to pay up to $4.85 billion in settlements. In 2011, they paid another settlement for $950 million, and just last year, they settled for $23 million. This entire scenario could have been avoided, as is shown by an article published in the Lancet Journal in December of 2009. The authors simply read, they studied the risk of cardiovascular events in Refecasive, and they did this by simply reading the bibliographic literature. They read articles from the bibliographic databases and some files from the US FDA. And their experiments showed that by the end of the year 2000, there was certainly no evidence to suggest that the faulty results could be attributed to the control. And so this is a classic case of what we know to be literature-based discovery. Literature-based discovery, it refers to the use of papers and other academic publications to find new relationships between existing knowledge. And that's the focus of this dissertation. It is a field that was founded by Don Swanson in 1986. Swanson, in much the same way like the folks did in the Lancet Journal, he had read the titles of over 4,000 articles on Raynaud and fish oil. And by looking at intersecting terms in the titles, he was able to find 3 unknown connections between Raynaud and fish oil, which have now been published and is a staple part of that research. Swanson formalized his observations as what we now know as the ABC model for literature-based discovery. Essentially what he says is, if we are attempting to make a discovery between two things, we're trying to find new knowledge, if we can develop a system that will find unknown B terms between A and C, then these B terms are candidates for discoveries. The research in this area has progressed towards richer representations beyond just ABC. Early research used keyword-based, concept-based, relations-based, and ultimately graph-based approaches, and some hybrids. Of significance among these is the approach by Dmitry Ristovsky in 2006, published in M.H.A, in which he introduced this idea of discovery patterns. Essentially, using structured background knowledge, what Dmitry says is, if I know that one class of things participates in some specific relationship that affects another class of things, and there's another relationship that's opposing the previous relationship, then I may be able to say that A maybe treats C, that sort of thing. The issue here is, of course, that there can be longer associations between A and B, which was noted by Bart Wilkowski in M.H.A in 2011. We had a paper last year in JBI that also illustrated the same thing for recovering Raynaud and fish oil. What our JBI paper also showed, however, is that not only do we have these A and C, or ANTS connections, paths essentially between concepts, but in terms of elucidating and really understanding the deeper meaning of how things are connected, one might be able to create subgraphs where several of these paths come together based on context. And this is the basis for the context-driven subgraph model, which is the first contribution of this PhD dissertation research. So just to give some more specifics before we move forward into details, our JBI paper was of some significance because what we showed when we moved to the subgraph model, and here is the intermediate platelet aggregation that Swanson found, not only could we recover the primary association that dietary fish oil disrupts platelet aggregation, which causes Raynaud, but we also saw that these fish oils, salmon, mackerel, and so forth, they stimulate or they produce these prostacyclins or epiprostenol, and it is the epiprostenols that actually treat the disease. In the process, the epiprostenol, the prostacyclins, they disrupt platelet aggregation as the mechanism for doing this. We did this by putting together manually these subgraphs after we extracted out using the tool for triple extraction from NIH, SEMREP, developed by Tom Reinfleisch. After we extracted out those triples, we manually put together the subgraphs, and this is what we were able to see. This was important work because we had been unaware of any other technique that went into this level of depth and detail in terms of rediscovering and decomposing Swanson's association. We subsequently learned that one of our Noesis alumni, Dr. Karthik Ramakrishnan, had in fact done this, albeit automatically. Unfortunately, Dr. Ramakrishnan's work is unpublished research, and so it's not visible to the scientific community. And so we are unable to do a thorough comparison with that work. In any event, the finding these complex associations is only one aspect of our research. What we found is that not only are there these complex associations between Raynaud and fish oil and so forth in terms of platelet aggregation on the dimension of cellular activity, but there are others on the dimension of blood physiology with blood viscosity and lipids and so forth, with vasoconstriction in terms of tissue function. And so the question for us, fundamental question that we address in this dissertation is not only how do we create these complex subgraphs, but how do we capture context so that we can create these subgraphs on multiple thematic dimensions? How do we automate this?
So here's a question. You have this tool audio. Could you choose to find the, think of it as you have these three views of interrelationship between dietary fish oil and Raynaud's disease. So could you choose the context independently and study the relationship across each of these contexts separately?
We don't provide functionality for that in the tool.
Dr.
Shet's question is, in the tool Obvio that we have developed, do we provide functionality to allow users to choose the context so that they can choose essentially how they view these complex associations? No, we don't provide any features to do that. We automatically generate the subgraphs and let the data essentially give us the various contexts. Now, that would be a good enhancement for future use.
So who would figure out that these are the three contexts? Is it apparent, or is it just through a lot of looking at the results that you can figure out there are three different contexts in which there are relationships?
One way to do that would be to actually label the subgraphs just to lead the user. But then ultimately, like I said, it's the human that would need to go and take a look and determine what the context is.
The thought is the following. If you look at Go ontology, there are three key parts of the Go ontology, right? Cellular functions and biological processes and so on.
Sure.
So you know that there are three important things as an example. So one approach could be simply to focus on each of those things. In your case, maybe if you're talking about disease, then there are, these three things are routine, I suppose.
Well, right. I believe that if we provide functionality for users to filter the paths that get included in the subgraphs during the subgraph creation process, that will only be a way to refine and allow the user to drill down. We just haven't provided that kind of functionality at this point. But it would be an enhancement for the future. It's the kind of thing to look at in the future.
I have one more question,
Sure.
so just go back to the previous one.
Sure.
So is platelet aggregation responsible for increase in blood viscosity?
Oh, no. We don't know that to be the case. As far as I know, no. These are, no, as far as I know.
So these are actually independent things? Like they're not kind of causing at the different level of detail?
Not necessarily completely independent, Here we see the prostaglandins, they actually disrupt platelet aggregation, but they also disrupt vasoconstriction. So they have a dual function, essentially.
So basically all of these show up in the result. Now whether platelet aggregation and blood viscosity are they're both affected, but how they are affected, and that may be a separate thing.
That may be a separate issue.
You'll have to further do investigations
I'll
on that.
talk.
It may be interesting to do. But yeah.
Yes, I'll talk later on about what I've called dimension context, where one might be able to reason across these substructures. We haven't done that, but that's the kind of thing that I've thought about. All right, so just to move forward, so the task is to automatically create these complex associations on multiple thematic dimensions based on context, and this is the basis for the context-driven subgraph model. Now, this is an important problem. To use context is very, very important, because the main issue when trying to address this problem is the issue of combinatorial explosion. This is the graph for Alzheimer's and arginine, all of the literature on Alzheimer's, all on arginine, all of the triples, put it together in a graph. This is a complex labyrinth, it's unintelligible, one can't make sense of this. The subgraph model essentially would like to take this graph of predications or triples. From that, given two concepts, extract out a more meaningful set, a more promising set from which discoveries are likely to arise. And that would be paths between Raynaud and fish oil if those are my source and target. And then from that candidate graph, use context to partition the graph and ultimately arrive at these subgraphs on these different thematic dimensions, ideally where no two contexts are the same. The question, of course, is, well, how do we define context so that we can be able to do this? And that's what we'll talk about for the remainder of the talk. To specify context, we derive foundations from both linguistics and semantics. In particular, we believe that we can represent the problem of automatic subgraph creation as one of capturing the relatedness between paths. Is everyone there? Is there any issue? Can everyone hear me just fine, or?
Yes, yes.
Okay, right. So we believe that we can represent this problem as one of computing the relatedness between paths. That would require that we specify the notion of the context of a path. Now, since we know that a path consists of a sequence of semantic predications or a sequence of triples, then the problem can be decomposed into how do we capture the context of an individual triple. To do this, we make several observations about the characteristics of Medline articles, which is our main corpus. One of the things that we observe is we know that Medline articles contain provenance metadata, the title, the date of publication, the authors, affiliation, and so forth. We know that they also consist of semantic attributes, and these are, I'm referring here specifically to the M.H.A descriptors or the medical subject headings. These are terms that are assigned to Medline articles first by the medical term indexer, and then refined by humans who read the entire content of the text and refine this list in agreement that indeed the article is talking about some particular topic. Then there are supplementary concept records. I'm not sure the exact mechanism by which those are assigned, but they do also capture some semantics of the article. Then there's a second level of characteristics that we observe. Then these have to do with levels of abstraction. The textual content of the abstract, in essence, it captures a summary, a semantic summary of the meaning of the entire article, and this is true for abstracts in general. This specific article is talking about icosapentaenoic acid, or EPA, which is the ingredient in fish oil, potentiating or stimulating the production of prostacyclin-like material, which is epiprostanol. This happens when EPA is used together with arachidonic acid, as opposed to arachidonic acid being used in isolation. Well, there's a sentence in the abstract that conveys exactly this. The author says, finally, the perfusion with HbA, which is human albumin, it's a serum essentially, containing 10 mmol of arachidonic acid plus 10 mmol of EPA, resulted in a significantly greater production of this prostacyclin-like material than when the HbA was used alone. And so the fundamental point that's really being conveyed here is that EPA, it stimulates or it potentiates the production of prostacyclin. Now, that's captured in the abstract. If we look at the M.H.A terms that are assigned to the article, well, we see there's arachidonic acid, there's icosapentaenoic acid, there's epiprostanol, which is prostacyclin, there's humans, there's umbilical veins. These M.H.A descriptors, at a higher level of abstraction, albeit, they do speak to the meaning of the content. And then we see the semantic predications, or the triples, that have been extracted from the content. Here, the first triple in particular says that icosapentaenoic acid stimulates epiprostanol, which is prostacyclin. Well, this is exactly what the article is talking about. So if these things are true, then what we can do is we can represent first the document in terms of well, of course, the abstract that's normally done, but we can also also represent the abstract in terms of this concept level semantic summary, which is the mesh descriptors. We may also then be able to represent it in terms of the semantic predications. Since the semantic predications and the mesh terms essentially, we're considering them to be equivalent, albeit not equal, then one might be able to represent the context of a semantic predication in terms of the mesh descriptors that have been assigned to the article. This is the basis for our first assumption, which is to say, it's the interchangeability assumption that the concept level and the relational semantic summary of a Medline article are in fact interchangeable. Specifically, what we like to say here is that a semantic predication can be represented in terms of the mesh descriptors. Now, your intuition is correct. When we go back to the article, what we're seeing here is that not all of these mesh descriptors are relevant to all of these semantic predications. Let's see, veins produce epiprostenol. Well, humans in particular, that doesn't really speak to that. Humans contain veins, yeah, but not quite. So what we do is we make a second assumption. We say that for each of these semantic predications, if we take the distribution of all of the mesh descriptors that occur in Medline for the particular predication, then that might be able to give us an overall context. This is in fact our second assumption. It is the context distribution assumption, which says that the context of a semantic predication can be expressed as the distribution of all mesh descriptors associated with all articles that contain that particular semantic predication. And these two assumptions are the basis of our representation of context, which then enable us to represent the context of a path, and then to compute the relatedness between paths so that we can then cluster these paths into subgraphs along multiple dimensions. This sort of thing, it is deeply rooted in distributional semantics, which is a field pioneered by John Rupert Firth. Firth is famous for being one of the first people to talk about the context-sensitive nature of meaning. Essentially, he says, in order to understand a word, one must understand its context. The word bad, for example, can mean exactly that, or it can mean good, depending on the context. So what he says is, you shall know a word by the company it keeps. This is a fundamental idea that's used in information retrieval and vector space model and so forth for finding words that are similar. Essentially, the idea is that linguistic items with similar distributions are in fact similar. We are extending Firth's idea here to say that semantic predications or triples that have shared context in their distributions are in fact related, because we're interested in capturing related associations in these subgraphs, related albeit within some context, not similar. If we created a similarity subgraph, then if someone knows one of the concepts in there, then they probably know all of the others. Right, so now we go into some details on how exactly it is we do this. Well, let's assume for the running example I have that was manually created in the JBI article, if we wanted to cluster together or determine whether or not these two paths should be clustered, what we would do is, we know that each of these two paths, they consist of a set of triples or semantic predications. We know that each of these semantic predications occur in some set of documents. We know that each of these documents have mesh descriptors, and we know that if we, in fact, we can aggregate the mesh descriptors across the semantic predications to abstract the representation of the path in terms of, well, a vector of mesh descriptors. And if we did this for both of these paths, then essentially the issue of path relatedness, it now becomes one of computing the relatedness between these two mesh vectors. Well, if we do this, this is a much more familiar representation of our problem. We can then apply the vector space model and all sorts of things around this that we're aware of. The specifics of how to do this is what I'll talk about at the core of this dissertation research. I wanted to point out that this is the second contribution of this research. It is this representation of the context of a path as this vector of mesh descriptors. Suffice to say here that these mesh descriptors, in as much as they are assigned to Medline articles to index the content, they're also part of a hierarchy of background knowledge. And this, I believe, will be key in enabling us to actually capture both implicit and explicit context for the purpose of subgraph creation. Okay, so now we get into some details. Assuming here is an anecdotal example that the context of the path PI is as shown in this example. M.I, that's an abstraction for a mesh term. The numbers here represent the frequencies of these mesh descriptors across the paths. Well, so what we're seeing here is essentially we have two sets of mesh descriptors with their frequencies for two paths. Ideally, if we'd like to use a vector representation, cosine similarity strikes us as something that we might like to use. There is, of course, an issue here that cosine similarity allows us to compute similarity and not relatedness, which is what we'd like to compute. But if we did this, if we attempted to use cosine similarity, what we would need to do, of course, is represent the, and now my arrow here shows that I'm switching to the vector notation as opposed to the set notation. We need to represent these two sets on the same dimensions. And so all of the out-of-context things would be represented with zeros in the first vector. In the second vector, all of the out-of-context ones would also be zero. And then we would compute the dot product, and we would divide that by the length of the two vectors. Several important things strike us when we attempt to do this. First, what we see is that all of the out-of-context vectors in the first vector that are not in the second, these would not contribute anything to the relatedness score. This works fine for similarity. That's what cosine similarity does. Second, the same thing happens for those in the second vector, but not in the first. The more critical issue, however, is that the lengths of the vectors, the lengths of the out-of-context vectors, could, in a way that we would not like, lower the overall relatedness score between the two paths. And that would happen here for all of these out-of-context things. Essentially, it lowers the relatedness score between the two vectors. While this is good for similarity, it is not what we would like for relatedness. The truth is that these two paths, they are as related as their shared context. The descriptors M1 and M2 are common to both of these paths, and so that is their shared context. And so we approach this task of computing the relatedness between pairs of paths by using this fundamental idea. First, what we would like to do, we have two objectives. One, we would like to maximize the weights of these in-context descriptors, and then minimize the weights of these out-of-context descriptors. There are several ways one can do this. The first thing one can do is, well, let's take a second look at these frequencies. We can imagine that if we're just, well, let's say we remove the denominator because those are the lengths and those are bad news. But if we still keep the frequencies, it means that if we have a third vector that has M1 and M2 with different frequencies, then the dot product of these frequencies will give the two pairs of paths different relatedness scores. Well, this is also undesirable because, again, the paths are as related as their shared context. So the first thing that we do is we binarize the vectors, that is to say, we eliminate the frequencies. And we simply consider a mesh descriptor as either present or not present. This then, in my simplistic example, will reduce my relatedness score to be the dot product of the binarized vectors, which is in fact equal to the intersection of the two sets, which is just simply 2. Now, of course, our intuition is correct. The problem is not this simple. And just to make that clear, let's take a look at some of the mesh descriptors I've assigned here just anecdotally to make the point. Let's look at the first mesh descriptor. That's platelet aggregation, which seems to be present in both sets. Epipersonal also present in both sets. However, let's assume, well, let's take a look here. We see that platelet activation is present in the second vector, but it's not present in the first. Now, what we know from the mesh hierarchy is that platelet aggregation and platelet activation, one of them is the parent of the other. We know that as well for platelet adhesiveness. The platelet adhesiveness and platelet aggregation, they're siblings. So it seems to suggest, and same thing for epiprostanol, it's a sibling with the prostaglandin. It seems to suggest that these two paths are in fact more related than their exact matches suggest. One must take into account not just these exact matches, but also the inexact matches. And to do this, we obviously would like to rely on the structure, or at least that's the point I'm making here, that we rely on the structured background knowledge to provide this additional semantic enhanced shared context. Now, in terms of specifics, how do we leverage this in a meaningful way? Well, what we would do is, well, it's essentially the cross product of the two sets. And now we probably back away from the vector notation because the vector representation will really not help us all that much. And I'll talk about why I've done that a little bit later on. What we really would like to do is to take the cross product of these two sets, and for every pair of mesh descriptors that are above some threshold of mesh semantic similarity, then we would like to increase and increment our overall semantic relatedness score. And, well, we do that by using dice similarity as the metric for semantic similarity. We're making no novelty contributions here. In fact, I had Swapnil and Nishita, who work with me on this, implement the set-based approaches, that's Dice and Jacquard. They implement the path-link-based approaches, that's Rada, Wu Palmer, Leacock Chodorow. They implemented the information content-based approaches, that's Resnick and Jane Conrad, and there's one other. The actual threshold or metric that's used to select the threshold is not so significant as the observation that one needs to use this structured background knowledge to do that.
I have a question.
Sure.
So basically what you're saying is that you're not going to use count of the same thing appearing, but if related concepts appear, then you want contributions from them.
Yes, we would like to capture the relatedness between exact matches and inexact matches. And to capture the relatedness between inexact matches, we will use structured background knowledge.
And aggregation and adhesiveness, they mean different things?
They're very related, very, very related. Platelet adhesiveness and platelet aggregation. I know they're not the same thing, but they're highly related.
One close together. So one thing that I think, I mentioned this in a couple of our discussions in the past, is that I think you started down the right road here and then missed one opportunity in my mind, which is to say that assume that you have two related, you have a mesh term, and it's the only mesh term that's shared in the context. Mm-hmm. And it's perfect relationship. The mesh term is in both. If that mesh term is at a very high level of the mesh hierarchy, you have less related documents to my mind, or less related predications than if it's at a low level. So two documents that are about humans are not as related as two documents that are about cell cycle regulation in humans.
Yes, absolutely. And this is using the measure of specificity from the structured background knowledge to be able to speak to the relatedness of context. It's something that we've discussed. I haven't leveraged it in a specific way here, but it's something that we've discussed perhaps as an enhancement over this score. Right. One certainly can do that sort of thing. Now, on the other side of that, in the context of the problem, right, someone may say, and we've seen this, someone may say in the literature that several proteins, and then they mention selenoprotein, right, affect some very specific thing, but they don't mention like very specific things about the protein or the selenoprotein. And so what is advantageous for us at this point is being able to capture things at different levels of specificity, but being able to capture the mesh descriptors that actually talk about them as well. So, yeah, two sides of the same thing. Right, so yes.
And I'll finish off the question by mentioning, I think someone may be breathing into the mic, so people on the phone for 2 minutes, that would help the rest of us hear Deborah's presentation.
All right, wake up guys, I'm sorry, it's early in the morning. So Bruin question is, am I considering the number of concepts on one side of the tree compared with the number of concepts on the other side of the tree when I essentially do this kind of computation? Inherently, this is sort of what DICE does, but I suspect that you're getting to a deeper point here. What DICE similarity will do is it will take all of the ancestors of a given mesh descriptor, and it will compare, in fact, it gets the intersection of the set of ancestors for one mesh descriptor and the set of ancestors for the other. I suspect you're getting to a deeper point, but I think this is consistent with what Dr. Raymer said, that one might like to go a little bit deeper, and this is one of the limitations of this work that I've pointed out in the limitation section. So we manually set the threshold, I think 0.75 or 0.8 For dissimilarity, what happens is if the pair of mesh descriptors, so let's say platelet aggregation and platelet activation, they have a similarity score above 0.75, then we simply normalize that score to 1. If the score is below, we make that 0, and this is consistent with my two objectives, which is to maximize the weights of the in-context descriptors and minimize the weights of the out-of-context descriptors. And doing this, then the overall semantic relatedness score, it becomes essentially just the cross product of all of these pairs of mesh descriptors that are above the threshold. In this case, it would be 5. The one here for the exact match with platelet aggregation, and another one for platelet adhesiveness, and so forth. This is the third contribution, and I think the most important contribution of this dissertation research. It is in fact the use of structured background knowledge for computing this shared context, or this semantic enhanced shared context between paths. An unfortunate side effect of doing this is that paths that are highly related, those are paths that would essentially be similar with many mesh descriptors, they end up with a very high score compared with others that are not so similar that have lower scores. And so we apply a log reduction just to reduce the disparity between these scores. Again, we're making no novelty claims here. It's just an aesthetic reduction of the scores. So that when we view the scores on a graph, it's reasonable to deal with. To do that specifically, what we do is we take one term in the first vector, we compute the relatedness with all of the other mesh descriptors, and then when we get that score, we apply the log plus 1, and then we sum across the two sets. So if we have this metric now for computing the semantic relatedness between two paths, then our next task is to actually use it to perform the clustering. I've selected the hierarchical agglomerative clustering algorithm as my approach for doing this for two reasons. One, HAC is an unsupervised algorithm. I don't need to specify the number of clusters, and this is exactly what I would like to do. I would like for the data to naturally align based on its context, as I have defined. The second reason is this HAC, it's deterministic. There's no random walks or randomization going on, so every time I run the algorithm, I essentially get the same output. To do this, just to make things a little bit more palatable, HAC starts off by putting each of the paths in the candidate graph to be clustered into a separate bucket. I would take the first path in the bucket population phase, and I compute the relatedness score between that path and each of the other paths to be clustered. Whichever paths are above the threshold of, now this is a threshold for path relatedness, right? I need to decide when to cluster two paths based on the overall score derived from the mesh semantic similarity using background knowledge and the log reduction and so forth. I'll talk about that in just a bit. And then I move to the second path and I do the same thing. And after I have completed this bucket population phase, then the next task is essentially to merge the buckets. One can imagine that this path, after I compute the relatedness with all the other paths, well, one bucket will be created with these two, for my example. When I get to this path, well, it's going to create a bucket with this and the first one. And so you need to merge minimally buckets that are exactly the same. Of course, one may suspect that there may be some buckets that are sort of related, but they may or may not have the same paths in them. We would also like to be able to combine those. And the metric for doing that, it's called the inter-cluster similarity that I'll talk about in just a bit. Don't worry about my slide numbers. There's 10 or 15 slides that I'll just skip over because they're part of experimental results. So in the bucket merging phase, I do the same thing. I essentially take one bucket and I compute the relatedness score with the other bucket in terms of, it's really the average of all of the pairs of paths in the two buckets. That's the intercluster similarity, and then I merge them. An important step here is one needs to be able to enforce this threshold for path relatedness that actually cascades for bucket relatedness. Otherwise, we may end up recovering the original candidate graph, which is not meaningful to us. And so the question that we will look at next is how do we specify this threshold for path relatedness? Once we've done this and we have a cutoff for our number of buckets, then we simply rank the buckets and we present those to the users. So just to do a summary here as a checkpoint, we are attempting to compute this threshold for path relatedness. We have represented our problem We know that the path is represented as a vector or a set, really, of mesh descriptors. We've used this business of semantic enhanced shared context that uses the mesh hierarchy, structured background knowledge. We've applied a log reduction. And we said, well, we will manually set the mesh semantic similarity, and hopefully I'll get a pass for that. But we certainly don't want to manually set also this threshold for path relatedness. That would not be a proper thing to do. So for the Raynaud fish oil experiment, again, that I continue to refer to as my running example, we had no idea how to compute this threshold automatically. And so we just set it manually. I tweaked around and I eventually saw, well, at this threshold, the graphs that were being produced, they were readable, at least I could look at them and I could see some things. But to do this in a more principled way, what I did was, for each, and I think there were 200 paths to be clustered, 200 paths that we manually put together, I think in 14 or 15 subgraphs reported in the JBI paper. What I did was, for each pair of paths, I computed their relatedness score, and I plotted it on a graph. And this is what we see from that graph. What we see here is, on the x-axis, I have the distribution of scores from 0 to 3.5, and now my log reduction comes in quite handy, because this is just my range. I could easily guess 3 or 2.5 or 2.75, and get things working pretty quickly. But what I'm seeing here is that around 1.75 or so of relatedness score, I have about 1,300 or so pairs of paths. It's 20,000, 200 by 200 would be about 40,000 path pairs. So 1,300 or so of those, this seems to be like a mean value, it almost looks like, right? What we're seeing is that less than 1.5, let's say, there's not that many path pairs. Above 2.5, there's not that many path pairs. But around this value of 1.75, it seems like there are many path pairs to the left and to the right on either side of the mean. And for those of us who know our statistics, this is reminiscent of a Gaussian distribution or a normal distribution. If we wanted to, if we sampled the heights of all people across the world, we'd say take 10,000 people from each country. And if we plotted that in a graph, what we would get is, well, most people are around about 5'7", 5'8". I think this has actually been done. There's few people less than 5 feet, let's say, and there's fewer, relatively speaking anyway, who are above 6'5", 6'7". But most people are sort of around this mean. And this is classic of a Gaussian distribution. So I actually computed the mean and the standard deviation of for three of the initial experiments, and I plotted the distributions, and this is what I got. Now, this is good to see because the data here approximates to a Gaussian, albeit that it's not exact. What we're seeing here...
Sorry. One thing I would suggest actually here is it wouldn't take much effort to add in if you actually did a Kolmogorov-Smirnov test or Anderson-Darling test for normality and see if it is Gaussian. Yeah,
I did chi-squared.
You did chi-squared variant.
Not very good. Okay. Not a very good Gaussian.
Okay.
But again, this is one of the limitations that I talk about. And this is exactly what I'm getting into. What we're seeing here is that, well, it looks Gaussian, but not quite, right? What's happening is from 0, relatedness scores from 0 up to about 1.5, 1.6. Like, there's sparse data. These points need to be really close together. This actually makes sense in terms of the semantics of the problem. Each of the path pairs will start with Raynaud and start with fish oil. That means that each of the path pairs, well, each path, will have some triple that starts with Raynaud, some intermediate, perhaps some other thing, and then end with fish oil. Well, if you go back to where those triples have come from, which is from the Medline articles, then those Medline articles would have minimally been tagged with Raynaud or fish oil. And so there is some minimum relatedness score among all of the paths. It's certainly not going to be 0. I mean, if you took 2 paths that were just completely disjoint, then you would get some 0 score, maybe. But if all of your paths are on Raynaud and fish oil, you'll get some minimum score. One good idea that Varun suggested that I do here is I recompute the relatedness between the paths relative to the minimum or relative to the maximum score, essentially define the relatedness not just in terms of the actual relatedness, but also in terms of unrelatedness. And this is essentially what this will do, is sort of shift this graph to the left and make the points more compact and give us a better Gaussian. But in terms of have I done this in a systematic way, the answer is no. I have assumed that this is a Gaussian that could be good for our purposes and move forward in that way. So, assuming a Gaussian, we know several properties about Gaussian functions. One, Gaussian distributions have what's called these 2 points of inflection. At the points of inflection, one can think of them as demarcating some particular event or characteristic of the data. One could think of these as what it means to be short on the one side and tall on the other side in terms of my example with the heights of people in the world. An interesting observation for me was that values that were set across the 3 scenarios, and again, I had no idea about this detail of Gaussian distributions when I did this, I just said it to get good graphs. Well, these values were consistently between the 2nd standard deviation from the mean on the one side and the 3rd standard deviation from the mean on the other. Well, this gave me an idea that perhaps I could use the 2nd standard deviation from the mean as my standard threshold, and this is what I did. So given this metric now to compute path relatedness, right, so now we can cluster things together that are above this threshold, which is essentially the 2nd standard deviation from the mean, then we can now do our bucket merging based on standard metrics for bucket similarity. This is from the Christopher Manning textbook at Stanford from the Introduction to Information Retrieval course that Dr. Prasad has taught. And after we merge the buckets, then we simply rank them, and then we'll wrap up the presentation. Now, for bucket merging, single link similarity is not ideal. Single link similarity will essentially, for each, it will compute the maximum similarity score, and my dots here represent paths. The maximum similarity score across all pairs of paths in the 2 buckets. And if that maximum score is above my threshold, the 2nd standard deviation from the mean, then I merge the buckets. This is not good because it may mean that the other paths that are not the maximum, those may be fairly lower, much lower than the actual maximum. What will end up happening is I'll end up with these straggly clusters. Yeah? For complete link, which is the opposite, it will use the minimum similarity. If the minimum similarity score across all pairs of paths in 2 buckets, if that is above my threshold, then it must mean that all of the other paths are also above my threshold. Well, that will give me very compact clusters, which is not bad, but it's not ideally what I want. What I want are things that are related within this context, but not so related. I want things that are in the region. I want to find dietary fish oil with platelet aggregation, and I'd like to find, not necessarily platelet adhesiveness, that's too close, that doesn't tell me much. I'd like to find prostaglandins, which are of type icosanoid, and platelet aggregation is of type cell function. How it is that these 2 things are actually interacting with each other is very, very important. So if I use the group average, this will bias too much. It will take the intra-cluster similarity plus the inter-cluster similarity across the 2 sets. This will probably give me things that are too broad. What I've chosen is the centroid or the inter-cluster similarity, which essentially, for each pair of paths across the 2 buckets, it computes the relatedness scores, and then simply divides by, it's sort of like the average of the 2 sets. The final task then is to rank the clusters. To do this, I rank them by compactness, which is the intra-cluster rank. The intra-cluster rank simply takes a path, it computes the relatedness score with all of the other paths in that bucket and across all path pairs. Then it divides by n times n minus 1, and I get my score. An interesting thing that happens here is that when I take, let's say, 200 paths and I run this algorithm, I'll get some number of subgraphs containing more than 1 path. But I will end up with a number of singleton paths that did not cluster into any buckets because they didn't meet my threshold for path relatedness as I have defined. And so the question is, well, what do we do with these? Do we discard them, or do we find some meaningful way of ranking them and looking at them? To deal with these, I use the measure of association of rarity to rank them. Essentially, what rarity does is, here is a path PI. I take each of the distinct concepts in the path as a representation of the association, abstraction of that. So that's A of PI is an association, which is really just the set of concepts in a path. I fire a query to Medline, and I ask, how many documents contain all 3 of these things? And if I get back a value of 0, well, it means that these 3 things have never been talked about in the same Medline article. Well, these are things that we would like to look at because they may be candidates for making discoveries. The intermediates are not known. And so in this case, for singletons, because the size of the set here is 1, then this is really just the frequency of that association in medline. But I use the same idea to estimate and compute the overall rarity of an entire bucket itself, an entire subgraph that I'll talk about in just a bit. So just to summarize and rewinding down, I'll show the demo in just a bit. We have computed the relatedness between pairs of paths as a way of clustering subgraphs, or clustering paths into subgraphs, hopefully along multiple dimensions. We know that the path is represented as a vector of mesh descriptors. We have said that we've used this semantic enhanced shared context using this log reduction. We've manually set the threshold for mesh semantic similarity. The threshold for path relatedness is the standard deviation from the mean. For bucket relatedness, we know that there's a set of paths in a bucket. We've used the intercluster similarity for singletons. We've used the second standard deviation, the same thing here, to cluster the buckets, to merge the buckets, and then to rank the buckets. Ultimately, we use the intracluster similarity and this measure of association rarity. So I won't talk about the algorithm. You might have correctly guessed that there's a pre-computing step here where we compute the threshold of relatedness first before we actually begin the clustering. And we use that value to determine which paths will go into which buckets. The algorithm stops when the number of clusters in one iteration and the subsequent iteration does not change. I found that about 4 or 5 or so iterations, things settle down fairly quickly. Okay, so what have we done with this? Well, let's go back to our example where we manually created this subgraph reported in our JBI article. Again, this is a complex association that has platelet aggregation with all of these prostaglandins, where the prostaglandins disrupt platelet aggregation as the mechanism for presenting or treating Raynaud syndrome. Well, at the third standard deviation from the mean, like I said, I used both just for testing. This was the first experiment that I ran. I generated just one subgraph. And this is that subgraph. I generated 4 subgraphs at the second standard deviation. Well, here's that subgraph. You may not be able to see. What we're seeing is the fish oils, the concept fish oils, and icosapentaenoic acid, which are tantamount essentially to the same thing. And I'm seeing Raynaud phenomenon in Raynaud disease, paths that begin and end with those. Now, this is not surprising. This is what we would have specified as input to the system. What we are, however, seeing is that in this subgraph, there's also platelet aggregation with the disrupts relationship, as we had manually put together. And we're seeing that the set of prostaglandins that were here, well, we have the triple that says epiprostenol is a prostaglandin. We have one here that says alprostadil, not epiprostenol, disrupts platelet aggregation. Well, alprostadil is PGE1, epiprostenol is PGI2, I believe. These are all in the prostaglandin family. Now, your observation is correct. Well, you know, there are links here that are missing. And indeed, there are, right? And now this goes back to a fundamental point that I made in the beginning of the talk, where I said that the purpose of the computational system is not to actually make the discovery. Rather, it is to find these promising links and present it to the scientists. In 1986, when Swanson explored this research question, there were 4 articles in Medline on Raynaud and fish oil. There were no articles on any of these things. If you had given a subgraph like this to him at that point that suggested that, well, it seems that platelet aggregation are being disrupted, and epiprostenol, it seems like those are disrupting the platelet aggregation, and that might be treating it, that could save, in my estimation, the domain scientists quite a bit of time in terms of their exploration. Now, to make this a little bit more palatable, and I'll skip over these slides, let me go.
Let me just clarify, so in the previous slide,
Yes.
the left part, the left op is manually done, and the
Yes.
right part is...
This is automatically generated, yes.
So given this, you could read the appropriate literature and try to fill in the detail?
And that's where we will go now to the web application. I'm going to switch away from the full, let's see here. I'm going to go to my demo. And what my demo, I'm going to actually switch gears here. I'm going to show for magnesium and migraine, just to give an idea of the importance of having these things on different dimensions. So here I'm showing a process for magnesium and migraine where Swanson found 11 connections between them. If you're generating a single subgraph and you rediscover all 11, then you'll put them all into this one subgraph with all of these different contexts. The domain scientist might not even look at it, even if you did it. Right? My subgraph number one, what I'm showing here is that in 1986, and my thing is cut off here, I've cut off all of my experiments to only the data that Swanson had at the time. There are only 7 articles that mention both Raynaud and fish oil. There's one article here that at the time was published in 1973, some 15 years before Swanson did this, that actually suggested, it's an article in German, that, well, you know, magnesium glutamate, which is a neurotransmitter, I think, might be used to treat migraine. And the article sit for 15 years, and that was it. Swanson found, and I'll show my first subgraph here, subgraph number one that I generated among 25 subgraphs shows that, well, magnesium interacts with serotonin, which is a neurotransmitter, it's of that glutamate family. And it seems like serotonin causes migraine disorders. Well, for the domain scientist, okay, so if serotonin is causing, if that's true, migraine, and magnesium is treating that, then perhaps magnesium is disrupting or inhibiting the serotonin levels. Now, the triple that's been extracted here says magnesium interacts with, and so that doesn't really tell us much. On the more interesting side, for whether or not magnesium causes that, in my tool, I click on the arc, and I get a set of articles. I think I get here 4 articles. The second article shows me in quite some interesting detail. It says that much evidence has implicated serotonin in the pathogenesis of migraine, or in causing migraine. Well, let's look at that. The article starts off by saying serotonin, which is released from aggregating platelets, and I stop because I know that platelet aggregation is a second intermediate discovered by Swanson. So it says serotonin release can reach sufficiently high concentrations to affect local vascular functions in a number of ways. The monoamine can cause contraction of blood vessels. That's vascular. That's the third intermediate that was found by Swanson. I read 2 sentences. By providing both these complex associations with the provenance of the triples in the scientific literature, we have effectively created the system to facilitate, provides these promising links that facilitates this process of making discoveries. I have found 8 out of 9 existing scientific discoveries by using this framework. As far as we are aware, that is the greatest number of rediscoveries that any one single system has made by automatically generating subgraphs and then allowing domain scientists to look at the provenance. Now, some may say, well, you know, you could have done this using statistical frequency, you could have used pointwise mutual information, you have not shown that singular value decomposition will not work, or LSA. And yes, indeed, point taken. Since we have not used any of the traditional frequency-based techniques, graph-based techniques, one might think that, well, whatever we're doing, it's just a hack job, right? But interestingly enough, the technique that we've used has produced concrete results. Whether or not something else can outperform us or do just as good, there still is something to be said about the use of context from structured background knowledge.
So why would something outperform? I mean, what would be the basis of it? So there are several things going on here, right? I mean, one is, what are you considering as an input? You have your data, basically scientific literature, and you have your approach takes as an input XYZ. You have, of course, the text, you have the provenance information, you have the mesh descriptors, you have the semantic predication. So you have a bunch of input that goes into your processing.
Yes.
And then there are some techniques. So you describe some techniques, you made a number of simplifying assumptions, but nevertheless the technique centers around capturing relatively rich representation of connections between the data that you collected.
Yes.
And then you have, and that includes cutting off, accepting level, and so on and so forth. Well, I mean, when you look at any alternatives, are they taking more inputs? Are they taking richer representation? Have you shown richer representations gives you insight that they don't give? Is a simplified representation better? Why would there be a situation?
Well, I was trying to get my overflow sites to come up, unfortunately not. SVD, singular value decomposition, is one approach that could be used alternatively, I believe, in a meaningful way. And that's because SVD will rely on the actual distribution of the data in the corpus to be able to find things, substructures essentially, that are connected, albeit not explicitly. And it does this.
Would SVD exploit semantics behind match distribution?
The answer is no.
The
answer is no, not in the way that we have done.
I mean, how can you show that that is important and better? Because if that is what SVD cannot capture, and you can show that through an editor, then maybe you have a case.
I don't have a worked out example of that. And so this is why my thesis statement here, it says that our claim essentially is that we are simply creating an information processing system, right? That, I apologize for the issues. Essentially what we're saying here is that an information processing system that leverages these rich representations based on implicit and explicit context, they provide an effective means for literature-based discovery, and that is what we have shown. Whether or not SVD or PMI may provide a more effective means, we have not addressed that question.
People have used SVD already, right?
Right, but...
So have they shown more?
The answer is no. We are showing here complex subgraphs on multiple thematic dimensions with the relationships in the graphs, and also the provenance of the literature. We have simply not seen that kind of thing. Kartik's approach, where he creates one subgraph using a greedy algorithm, claims to have found Raynaud fish oil, although that is unpublished work. We have simply not seen someone provide this much evidence for a technique by finding complex things on so many different dimensions. And just to make that point, and then I'll wrap up, one might have, from my demo, I showed here with serotonin, which is a monoamine neurotransmitter. My second subgraph, and I apologize if my display here is not that great, screen resolution. The second subgraph contains magnesium and water and control groups and so forth, which is different in any case, although the subgraphs are not labeled, from what we see on serotonin. The third subgraph has clonidine, magnesium and clonidine. I'll skip forward. I think there's subgraph, I'll go to subgraph 11, I think, that has glucose and insulin. Again, on the different dimensions, subgraph 15, that has magnesium, glucose, uric acid. There's subgraph 22 I can't get to. These things are on different thematic dimensions. I'm not really seeing the same information across each of my subgraphs. And so using this particular approach, I'm able to find 7, I think, out of the 11 associations Swanson found. And I'm not really finding, well, for serotonin, I get to 2 or 3 other things from the text. But in the subgraphs, they're on fairly different dimensions. So just to wrap things up, the time is running away from us. Skip all the way. Okay, so just to wrap things up here, again, I apologize for switching between screens. We did a statistical evaluation to estimate the interestingness of the individual subgraphs. And essentially what we did is for a given subgraph, we go to Medline, we compute the rarity score of each of the associations, and we do this across all of the paths in the particular experiment, and then we divide by the total number of paths, and we get an overall rarity score. Let's say, I'll just show the results to show that. For Raynaud and fish oil, we know that in 1986 there were only 4 articles that mentioned both of them, obviously no articles that mention Raynaud fish oil plated aggregation. So we're not surprised by the number 0. This gives us an interesting score of 1. For testosterone and sleep, the discovery made by Tom Rindfleisch and Chris Miller, there were 654 articles that contained the unique associations. That's an average of about 10 or so articles per association. And so these are not as interesting. I would go back to my demo, but we have found in an individual subgraph for testosterone and sleep the exact information that Tom and Chris Miller showed. This is quite interesting stuff, I believe. So just to wind things down.
I have a question.
Yes.
So you kind of set aside one group called association rarity or something of paths that were below the threshold, right? I mean, when you were trying the clustering, you had one bunch of paths that didn't quite cluster well because they were below the threshold for all the cases, and so you kind of separated them out.
Yes.
So is that the place where things will work out nicely for interestingness score? I mean, is that
it's actually
is that the
across
target
everything.
for this?
It's across everything. I simply take everything and I compute the interestingness score of all of the subgraphs, of just all of the paths in the experiment. Just as an idea of if randomly, without looking at zero rarity things and so on, if we presented an expert with a subgraph, would they even look at it? Would it be interesting in general? And it seems to suggest that perhaps they might. In my web demo, we're showing the integration of the predications graph with the corpus on the one side, and I haven't shown on the other side, if the graphs become too large, you can actually click the semantic types to filter them down. I'm actually running out of time. This is the fourth contribution here. It's not a deep integration, but it's this idea of combining the assertional knowledge with definitional knowledge to facilitate literature-based discovery. Last thing, I've talked about context a lot. I've talked about context at the level of semantic predications as a vector of mesh descriptors. I've talked about context of a path by aggregating these mesh descriptors. Talked about the context of the shared context of a subgraph, or shared context of two paths, by aggregating the mesh descriptors and then going to structured background knowledge to get inexact matches. And then I've talked about the context of a subgraph, although the context of the subgraph is not captured through a label. And again, that is being obtained through mesh descriptors above this threshold. There's this idea of dimension context, where we have subgraphs that have these disparate themes. If one can reason, as Sutton did for his experiments on cell division, linking that to the experiments that Mendel had found on plants, if one could find a way of reasoning across these substructures, that would be a really nice thing to do. That's the kind of stuff that I stay up at night and I get very excited about. We haven't addressed it here, but this is certainly the kind of thing to look at in the future of this kind of research. It's this idea of reasoning across these dimensions. I've made 4 contributions. One, the context-driven subgraph model, and I've shown earlier that you could use this to rediscover and decompose Swanson's association. We've automatically created subgraphs on multiple dimensions, and we found, I believe, 2 out of 3 of the associations with complex information there. We've specified this business of predication context in terms of this vector eventually downgrades to a set of mesh descriptors. We've used structured background knowledge to compute the shared context. And then we have, in my web application, which I didn't show, we've used both the literature and the background knowledge to be able to filter things. Concretely, this work is distinct from all of the other things that I am aware of in one sense, if not any other. We are the only people to have done work where we are automatically creating these subgraphs seemingly on different themes. There's work that automatically creates subgraphs. Some of them have relationships in them. Some of them provide evidence from the scientific literature. But I'm simply unaware of an approach that creates subgraphs on so many different dimensions that has, in fact, been able to recover so much information. So we have some degree of confidence that if we were to apply this to all of Medline and to all of the concepts in the UMLS and create a Google-style interface where there's a drop-down list when someone types something so you can search. So essentially what this would be, it would be a different way of indexing the Medline corpus, not based on the documents that are there, but rather based on the atomic corpus. This, we believe, is something that could be quite significant. Again, we've used it to rediscover 8 things. Several limitations. One, the manual threshold for mesh semantic similarity, not cool. Right? There's many things one can do there. The threshold for relatedness, our data approximates the Gaussian, but not quite. I mean, you could do some better things there. Our definition of context uses just the mesh descriptors, I had a very intense discussion with someone who felt very strongly that relationships could be used as well. In the end, we selected the M.H.A descriptors because, well, they're manually assigned to the articles, and we believe that they should capture some kind of a concept level semantic summary that can be leveraged as we have done. But again, there can be an ensemble of approaches, ensemble approach where several features are being used. That's possible. We're not doing a deep integration here of the background knowledge of the scientific literature. Our focus here really is on the literature, not so much on the background knowledge. If someone's making discoveries from background knowledge, there are several issues. One, unless domain experts put the knowledge in the background knowledge source, well, you're not going to find it. If you did a search today on Medline for P53 and cancer, and you did a search in the UMLS for P53 and cancer, there's an obvious disparity between all the information that's there that domain experts just did not know and put into the UMLS. Now, you may be able to discover things from the UMLS, but that's ontology-based discovery. We're not in the ontology-based discovery business. We're doing literature-based discovery. Suffice to say that I believe that it is the combination of these two things that will be most effective. But our work is, again, the title of my dissertation is a context-driven subgraph model for literature-based discovery. Contradiction detection is something we haven't done. There are contradicting predicates in the graphs. We simply allow the domain scientists to go to the literature and to sort those out, consistent with my argument that it is the domain scientist that will make the discovery, not so much the system. A better statistical evaluation is perhaps in order than the one that we've done, and I think the most important thing is the scalability of the clustering algorithm. It runs at about 45 minutes. We just got a new machine while I'm on my way out, but it would be interesting to run this on all of the UMLS and provide this new way of indexing the biomedical literature. It is an open secret that my long-term aspiration is to get into entrepreneurship. Perhaps this is the kind of thing I might be, may or may not be successful at, we don't know. Labeling the subgraphs could be very helpful, although we've seen that it's not absolutely necessary, but it certainly could be helpful. So the future of information processing systems, we believe, could and will benefit from richer representations to be able to extract the knowledge and to mine and analyze the knowledge in both structured and unstructured text. We have seen this in personalization. We've seen the use of structured background knowledge to create hierarchical interest graphs. IBM has a project called BlueMix that's doing human activity modeling. We've seen this in mobile applications. We've seen this in our prescription drug abuse project where we are mapping slang expressions to structured background knowledge and using that to facilitate search and text exploration. We believe that leveraging such rich representations will be able to leverage together implicit context with explicit context and ultimately move towards these powerful semantics for, in fact, various applications. This dissertation simply applies this idea to literature-based discovery or to biomedical literature, but the fundamental idea remains, we believe, strong for a variety of applications, which we have seen. Don't need to repeat, we have used all of this to rediscover 8 out of 9 things. I've published a number of things. Most of my research is on text mining and literature-based discovery. I've done quite some work on the pre-dose project, and I thank Dr. Carlson for his attendance and for working together. I think the pre-dose project has been one that we've been very pleased with. I leave you with this, this quote from H.P. Lovecraft, who is a science fiction writer. Lovecraft says that someday the piecing together of dissociated knowledge will open up such terrifying vistas of reality that we shall either go mad from the revelation or flee into the deadly light and into the peace and safety of a new dark age. We are all progressive folks, and this is one of the times when we would like to move towards the light as opposed to fleeing into darkness. I thank you for your time. I thank a number of people who have assisted throughout my PhD. Of course, my committee, Dr. Sheth, for his leadership and his guidance. I'm particularly grateful to Dr. Sheth for always challenging me to address real-world problems with real data, to come up with real solutions that have a real impact on society. Dr. Raynaud for many insightful discussions, some of which he raised today, which we still have not done, but I think which enhance and put into focus various aspects of this work. Dr. Prasad, for many discussions we've had, not just on this research, but on several others, and for always getting down to conceptually what's happening and forcing me to put things into perspective. I thank also Dr. Reinfleisch. I did 2 internships at NIH. In my discussions with Dr. Reinfleisch were always intellectually charged, very stimulating. Dr. Reinfleisch has made 2 discoveries from scientific literature, and he continues to be a pioneer in the field of LBD. I thank also Dr. Kavaluru at the University of Kentucky, who is someone who is very focused on details, and I really appreciate those discussions, my notations, my formalism. I owe whatever I've learned there to him. I thank Varun Bhagwan, who was my mentor when I interned at IBM. I think I've learned from Varun this idea of dialogue with data. When solving a research problem, while it's important to know a bunch of techniques that may be able to solve your problem, ultimately one must take a look at the data. The data will actually tell you whether or not the technique that you're thinking of is appropriate, and one might be able to derive a more appropriate solution. And so I thank these folks and all of my Noesis friends and colleagues, those who are present, but those who are also joining us remotely, for quite a wonderful experience. I've learned a lot professionally, certainly being here at Noesis. I think I've learned more personally than I have professionally. So thank everyone, and thank you for your time.
Take questions for people not on the committee, and then go to the questions from people on the committee. So, yeah, okay.
Well, I have a question on, so we did just the way on literature based. What if we have the web pages, and the web pages put those tags? So we have a lot of these tags.
Yes.
In
The question Emmett is asking is, well, is your work applicable to just scientific literature? And this is a question that's come up before. In principle, in fact, I've had an interest in applying this to legal documents. In principle, everything holds. What you really just need is a way to index the legal documents, and keywords may already exist. After you index the documents, you would need some kind of a legal ontology, and I've actually seen some of these in circulation. But in principle, we can really apply this, and this is why I made the point that I believe future information systems will need to leverage with structured background knowledge to be able to interpret and analyze data in a variety of applications. So yes.
So
yes.
The interestingness score or something like that, right? You had something like that. So do you think there will be everything in the corpus that's probably not even related in the interestingness? How do you deal with such a scenario? Because there may be literature based like documents which may not mention 2 concepts because it doesn't make sense. Or something.
Well, right, and so our approach here is doing exactly that, right? It's taking statements from one document, let's say, it's taking another statement from another document, and it's essentially connecting those statements based on the shared concept, and of course the underlying mesh descriptors that are used for those documents. Okay. So while an individual document may, and we've seen this for testosterone and sleep, and perhaps I can show this now. For testosterone and sleep, Tom and Chris Miller, they had, the key piece of the puzzle was really what was happening in terms of testosterone and cortisol from the cortisol literature, and here's subgraph number 7, that shows, and this shows the exact relationship here correctly. But what they knew was that, well, cortisol disrupts sleep. That was sort of well known. And this is why we have this rarity score that's 10, interestingness score that's very, very high, or very, very low, the rarity score very high. The critical piece was what's going on with testosterone and cortisol. And in my application, it turns out that it is this first article by Raynaud that is actually cited in the sleep paper as the article that gave the key information. So this is why I actually did not implement PMI or SVD or these kinds of things. Fundamentally, I do not really believe that, that it is frequency of distribution that speaks to discoveries or speaks to new knowledge. It is context that speaks to discoveries. On the one side, cortisol and sleep was well talked about. On the other side, cortisol and testosterone was not really all that well talked about. What brought them together here is their context. Testosterone inhibits cortisol, and cortisol disrupt sleep.
So again, when you say SVD, you're talking about comparing documents, right?
Well, you could represent the path in terms of the M.H.A descriptors, right? As a representation for like a term document kind of, or document term matrix. That kind of. So you could take a path, and my background slide is not coming up, but you could do that. Yeah. Okay. I think we'll wrap up here and then take questions from just the committee. So I thank you once again.